So I'm going now to talk about what is, I would say, my favorite topic or my favorite scientific topic. It's about how we can improve our biological control agents. Talking about biological control agents, I work mostly with parasitoids. And many parasitoids need, many, no, all of them, I would say, need to feed on carbohydrate sources for long periods. And this is something that you can see in this graph for the students. Again, I'm repeating this graph. I hope you don't mind. And this graph here is telling us that a parasitoid with food, in this case honey, can survive for more than 40 days. But this same parasitoid, without a carbohydrate source, will die in just a couple of days. So that means if we have a nice biological control agent, a nice parasitoid, or also a housefly, without food, it's useless. From the point of view of biological control, it's useless. It's not going to parasitize. It's not going to prey upon our pests. And the main carbohydrate source in agriculture, it has been always considered nectar. However, if we think on nectar, an agricultural crop, please keep in mind your crop, the crop that you used to have close to your house, the crop that you work on. Think how long it lasts the flowering period. How long lasts the flowering period of apples here in Emilia Romagna? A couple of weeks. In the case of cereals, they don't even have nectar, right? No nectar. Vineyards, tomato. Most of the crops have flowers only in a few weeks, or they don't even have nectar. So nectar, actually, in agriculture, is a poor source. In citrus, for example, that is my crop, the flowering period lasts 3 weeks. The rest of the year, where are the flowers? Do you see flowers? No nectar. So the parasitoids cannot find a carbohydrate sources in agriculture. It's very hard because we have prepared our crops to have no flowers. This happened in citrus, but as I said, if we move to cereals or other crop plants like soybean or potatoes, then we cannot find flowers either during most of the crop season. However, if we look close to, I'm sure that in all these crops we will find aphids. Aphids, whiteflies, millas. There are always a few emitters excreting honey, and it's happening cosmically. Always, if we look in some plants, there are so many emitters excreting honey. And honeydew, like nectar, is a rich carbohydrate source. This is well known. If you ever touch honeydew, it's like caramel. It's a sweet. Okay? So it's just full of sugars. In fact, in citrus, as I said, we do not have flowers. But look how many honeydew producers we have throughout the year. In the spring, it's full of aphids. In summer, we have the mealybugs, as I explained to you before, soft scales, also Etheria burgessium. And at fall, we usually have whiteflies. Same happens in all crops. But, okay, we have honey. Who is feeding on honey? Do parasitoids feed on honey? Do bees feed on honey, or only ants feed on honeydew? That was unknown. Well, it was known that ants feed on honeydew, of course, we know. And also, in some areas, know that bees feed on honeydew. In fact, there are honey made from honeydew. This is typical, for example, in New Zealand, but also in Greece, also in Italy here. I didn't know that. So some honey is made from bees that feed on honey. Okay? Also solitary bees feed on honey. But what happened with our biological control agents? Do they really feed on honeydew? Do these small wasps look for honeydew? So to answer that question, a few years ago, we had a very interesting parasitoid. It's called Aphytis melinus. This parasitoid attacks the armored scales that you can see here. Armored scales do not excrete honeydew. So that means that this parasitoid has to look for either to attack the scales, the armored scale, or look for food. So they need to feed on a carbohydrate source, right? So does it feed on honeydew from other pests? So that was our question. And we went to the field, measured how many honeydew producers we had in the citrus. And as you can see, the spring was full of aphids. At summer, we had mealybugs that were splitting honeydew. But at fall, the number of honeydew producers was much lower. Then we measured the different sugars that we had in the honeydew. Look, that's quite interesting. And I'm sorry for this huge table, but it's telling us the different sugars. You can see here different sugars: fructose, glucose, sucrose. I'm sure you know these sugars. And we check these sugars in the different honeydews. Look, we found two sugars called erlose and melesicose that are found only in honey. In other words, in your sugar, in your apples, oranges, in the sugar that you will eat in a cake, you will never find these sugars because you only can find them in honey. So if we have these sugars that are specific of honeydew, that means we can use these sugars to check whether our parasitoid is feeding on honeydew. And that's what we did. We knew now that these emitters are excreting honeydew with these two sugars. We came back to the field and captured the parasitoid that is 1 milliliter and measured with the HL, with the HPLC to check if it has any of these specific sugars. And with that, look what we found out. In spring and summer, most of the parasitoids have either feed on sugars or on honey. In fact, we are almost sure that this one had also fed on honey. But the amount of loss or melesicose was so low that we were quite cautious and we did not consider honeydew. But you can see that most of the parasitoids in spring and summer, when there were many honeydew producers, they were fed either on sugar or on honey. However, look what happened at fall. See here at fall, most of the parasitoids were either unfed or starved. They did not contain carbohydrates. They were not feeding, they were starving. A parasitoid like that may die in 1 day. Do you remember the first graph I showed you? Without sugars, in 1 day they are dead. These parasitoids were dying. In fact, what we did at fall is we captured the parasitoids at the beginning of the day, at noon, and in the afternoon. And we measured the amount of sugars. Look. They were losing sugars. They were dying during the night because they were starving. They are not feeding throughout the day. So we have demonstrated with this study that parasitoids feed on honey. Even if their host does not excrete honeydew, they are able to look for honeydew and feed on honeydew, a parasitoid that is 1 millimeter. Okay? And in fact, during the last years, not only in my lab, but in many other labs in the world, have demonstrated that many different natural enemies, very different from chrysophids, sylphids, parasitoids from very different families, feed on honey. So when they do not have nectar, they feed on honey. Okay. So we have demonstrated that natural enemies will feed on honey. So then, why in the past we did not consider honeydew as a food source? We only care about nectar. It's because honeydew was considered a poor food source. Is it the same to have a good diet for you, but to eat every day from a bad sugar? So it was thought that honeydew was a poor nutritional food source. In other words, if the parasitoids feed on the honeydew, they will die soon because this is a poor diet. That was considered in the past, especially compared to nectar. But just 9 years ago, our colleagues from Portugal compared the nectar and honeydew in olive trees. And they compared the survival of several parasitoids fed either on nectar or on honeydew from 2 different species. Look with gray color. This is called Seissetia oleae. It's the black scale, the olive black scale. It's this scale that you can see here with the Crematogaster ant feeding on the honeydew. And you can see that the survival of the parasitoid was much higher with honeydew than with nectar. So actually, honeydew can be a rich nutritional food source for beneficial insects, as our colleagues from Portugal demonstrated for this honey. In citrus, coming back to my system, what we saw is that actually the nutritional value of honeydew is highly variable. Some honeydew is excellent. The parasitoid will like for long periods when they feed on honeydew from mealybugs, for example, from Mithria purchasii. And when they feed on honeydew from this aphid, Apis spinicola, or from the whitefly, the survival is good. So what this result is telling us is that if we have honeydew, we cannot say it's a good food source for our animal or it's a bad one. We need to check. Okay. So actually, the nutritional value of honeydew is highly variable. And why is variable? First of all, 80% of the composition of honeydew are sugars. I explained to you at the beginning, right? Honeydew, if you touch it, it's just a sweet. It will be like a sweet, right? Liquid. And it's made of sugars that come from the plant, like glucose, sucrose, fructose. These are rich sugars, easy to digest. They do not crystallize. So these are considered high quality sugars. However, other sugars, like these ones, melissitose, cellulose, do you remember that I mentioned before? These come from the honeydew producers, and these are poor honey, that they are poor sugars. The parasitoids that feed on these sugars, they do not survive for long periods. However, the ones that feed on these sugars live for long periods. So that's one of the reasons why honeydew was considered a food source, because they contain many of these sugars. That was one of the reasons. The second reason, and was likely the most important why honeydew was considered a food source, is that aphids, for example, can feed on plants that contain toxins. For example, tomato.
tomato toxins for the aphids. So, for example, in the case of the milkweed, Asclepias syriaca, colleagues from North America check the cardenolians, and plants contain cardenolians. These aphids were feeding on the plant and were full of cardenolians. And the honeydew that they excreted contained these cardenolians. These are toxics. These are toxins. So this is showing us that honeydew can contain also toxins. And that's why honeydew was considered a good food. All honeydew contains toxins? Not all. But some of them may contain these plant secondary metabolites that reduce the nutritional value. That's why honeydew was considered a good food. But just a few years ago, in the lab, at that point was a postdoc in my lab, Pablo Paneja, checked not only the subparasite, but also the amount of eggs that the parasites contain. To produce eggs, you need proteins. And Pablo fed the parasitoids either with sugar, black points, with proteins, right? Sugar plus protein, or honeydew. Okay? And then he dissected the body of the parasite, cut the parasite, open the parasite, and count the number of eggs. These are the eggs actually of the parasites inside of the abdomen. This is one egg. This is another. Okay. You can see here that after 10 days with honeydew, the females that had fed on honeydew contain more eggs than when they had fed only on sugars, suggesting that honeydew contains also beneficial proteins. So some honeydew contain proteins that are toxins, we have seen before, but there are other honeydews like the ones that we can see here that even increase the number of eggs that the parasite contains. That means this parasite will parasitize more, going to kill more our pests. And that was quite interesting. Why does it have proteins, the honeydew? So we asked that. And what we did, we checked, and what we found was quite interesting. We measured the amount of proteins in honeydew excreted by the millibug, but also by the cyclic. And both of them contain proteins from the insect and from the plant. So in the honeydew, we could detect these proteins. We could detect the proteins in honeydew from the millibug, the cyclic, but also from many other insects. Many hemipterans, all the hemipterans are excreting proteins coming from the plant. And some of these proteins, or the most common one, is this one, superoxidase dismutase. This is an antioxidant. Have you heard about antioxidants? To keep healthy, to keep young. I guess you have heard that in the advertisements. So we found that some of these honeydews, most of these honeydews, contain antioxidants that are good proteins actually. So that could explain why the number of eggs in the parasitoids that feed on honeydew was increasing. So what we did is to feed the same parasitoid, remember the same parasitoid, but now with sugars, not just proteins, but this protein. Look what happened. The same that we found. So we found that this protein coming from the plant goes through the aphid. The aphid excretes it in the honeydew, and the parasite, when it fits, it benefits and matches more eggs. So this is telling us that contrary to what was thought before, plant proteins induced also by herbivory can have a high nutritional value for the parasite. So honeydew is giving us sugars for the parasites, but also proteins that can increase the egg. So I think this breaks the old paradigm that was telling us that honeydew was a poor food source for biological control. And this is showing us that honeydew that is very abundant in agriculture, because all the crops that you thought before, there are aphids, there are nectar secreting honey. And this honeydew can have a high nutritional value because it will contain good sugars and can sometimes contain also proteins that are beneficial for the parasites. Okay, so I have demonstrated one, that honeydew is abundant. Two, natural enemies feed on honeydew, even small wasps will look for honeydew and feed. Honeydew has variable nutritional value, but can have high nutritional value. But then is where our concern is, because I'm sure you know that the number of bees have reduced during the last year, right? It has been on the news that the number of bees have reduced, and one of the effects is because of systemic insecticides can reach nectar and pollen. This is one of the main hypotheses to explain the reduction of bees. So in the same way that honeydew is reaching the nectar, it may happen that systemic insecticides can reach the honeydew producers and the honeydew. That was our hypothesis. Can honeydew be contaminated with neonicotinoids? And to test that, we use the same system. We use the millibug, in this case, one parasitoid and one predator or pollinator, that is the circuit. And what we found out is that using different techniques, we demonstrated that the honeydew from these millibugs contain either imidacloprid or thiamectosan of the citrus trees were sprayed with these insects. But what really concerned us is that when we provided this food to our natural enemies, they died. When the circuit fed on honeydew coming from trees treated with imidacloprid or from honeydew treated with thiamectosan, as you can see, yes, most of the circuits died. And exactly the same happened to the parasitoid that fed on honeydew in trees treated with thiamectosan. So we have demonstrated first that the neonicotinoids can reach honeydew, but also in the right side, an amount that will kill the natural enemies that feed on this honey. That was published very well, and it was on the news. And because it was on the news, we received many critics. And we received two main critics. One was telling us that you are a parasitoid. Why are you going to feed honey that is contaminated? They may avoid contaminated honey, right? You are in a field. What would you choose? Honey contaminated or non-contaminated honey? That was the first critique. I accept that critique. It has sense. I will never eat something that is unhealthy. Well, I'm not sure. And the second one is that actually the most common use of neonicotinoids is not spraying them, is the seed treatments. So we accept that critics and we try to answer our critics. So for that, we came back to the system of the millibug and the parasitoid, but in this case, we used a choice test. We gave to the parasitoids either honeydew that was contaminated or honeydew that was not contaminated, and checked what the parasitoid can choose. So you can see here the response of the parasitoid Anagyrus to honeydew from untreated trees or honeydew from trees treated with thiamectosan. this graph is telling us that female parasitoids that were affected, have no previous experience with honeydew, were choosing honeydew treated with diameters. Why? We don't know. But almost 75% of the females chose to feed on honeydew that was contaminated. When they had experience with honeydew, previous experience, we fed them with honeydew that was not contaminated, then it was 50-50. It is that once they have fed on honeydew that is not contaminated, then they cannot choose, or they do not select them. Again, now we fed them first with honeydew with diameters. Again, they chose the one that was contaminated. So it seems that answering to the first question, the parasitoids prefer to feed on contaminated honeydew. But why? Why a parasitoid or us which want to feed on something that is contaminated? For smell. Smell better? So it is known that neonicotinoids affect not only insects but also bacteria. So neonicotinoids can affect bacteria. And in honeydew, we have many bacteria that produce volatiles. So we thought that maybe neonicotinoids may affect the bacterial composition of honeydew. And then the volatile, the smell. And in fact, we checked the bacteria abundance in honeydew, treated with thiamethoxam or not untreated in the control. And there were high difference. The bacterial composition is totally different in honeydew treated with neonicotinoids and in untreated honeydew. In fact, 6 bacteria were more abundant in honeydew excreted by millibags fed on citrus trees with thiamethoxam, as you can see here. Okay. So we think we have still not demonstrated, but we think that some of these bacteria can be responsible for the selection of honeydew that is treated. So they smell better for the parasitoid for any reason, and the parasitoid selected it. Well, we have already answered to the first critic. Parasitoids do not avoid feeding on contaminated honeydew. In fact, they select contaminated honeydew. And the second one was about the seed treatment. Is it really important, our finding for the seed treatment? I don't know if all of you are aware, but many crops, especially cereals, they are covered, the seeds, with neonicotinoids. And in fact, in the States, for example, most of the maize, the soybean, or the cotton, they use seed treated with neonicotinoids. And we checked that with the soybean in the fields of Minnesota with our colleagues from George University Lab. There, my colleague Miguel Calvo, that at that time was doing the PhD, checked exactly the same that we did with the millibag, but with the soybean aphid. And in this case, he used one predatory meat called Aphidoletes aphidimitha and one parasite that is called Aphelinus vector. And he tested seeds treated with thiamethoxam or with thiamethoxam plus fungicides in this case. Again, it's the same, and look, concentration of neonicotinoids in the honeydew. Non-treated seeds, we still could find some neonicotinoids. This is because everywhere, and in the class I explained to you, everywhere you can find them. Also, even if the seeds are not treated with found a little. But in the seeds coated with thiamethoxam, we found high concentrations of the neonicotinoid. And in fact, it was reducing the longevity of the Aphidoletes aphidimitha. This case was not directly killing, but it was reducing the survival of the predator. And the same happened with the parasite. So answering to our critics, again, soybean aphid excreted contaminated honey coming from the sea. And so far, since we discovered it 5 years ago, it has been demonstrated for 5 systemic insecticides, 4 immigrant species and 3 plant species that honeydew can be contaminated. So how this finding was important for agriculture. And just to conclude, I would like to explain that we should consider honeydew in conservation biological as a food source for our natural elements. Not only nectar is important, but also honeydew can be, in some cases, can be a good food source for natural elements. It will provide sugar and it will provide proteins, but we should be careful because some insecticides may contaminate. And with this, I finish. Thank you very much.